behalf of Cambridge Health Tech Institute's Breakout COVID-19 Global Web Symposia Series and our sponsor, Forte Bio, I'd like to welcome you to Rapid Characterization of SARS-CoV-2 Vaccine Candidates. My name is Elizabeth Lamb, and I'm the host and technical director for today's event. Now, I'd like to introduce our moderator for today. He is Nilshad Salim, PhD, Marketing Applications Manager with Forte Bio. Welcome, Dr. Salim. The presenter ball is yours.
Thank you, Elizabeth. Hello and welcome everyone to our webinar today titled Rapid Characterization of SARS-CoV-2 Vaccine Candidates using the Octet. And the webinar panel today includes myself, Nilshad Salim, Marketing Applications Manager at Sartorius, and I will give you a short introduction to Octet biolayer interferometry platforms before I hand over to our featured speaker, Dr. Wei-Hung Chen, who is a research scientist at Walter Reed Army Institute of Research, who will talk about his efforts on SARS-CoV-2 vaccine development. First, I would like to introduce Forte Bio's range of label-free biosensor-based instrument platforms. We have two main categories of systems based on their detection capabilities. The Pioneer FE and the Pioneer systems, equipped with next-generation one-step injections, are based on surface plasmon resonance, and the Blitz and Octet platforms are based on biolayer interferometry detection. In today's webinar, however, we will be primarily focusing on data obtained from our Octet systems. As you can see here, we offer a range of BLI platforms to choose from based on your throughput and sensitivity needs and also budget. Here, in the leftmost, we have the benchtop single-channel Blitz system. That is our lowest throughput system that is intended for large molecule binding analysis. Our K2, Red96E, Red384, and HDXs are the most sensitive BLI platforms that can detect and characterize small molecule interactions as well. The throughput of each system is governed by the number of channels in this system. The Octet K2 has 2 channels, the Red96 has 8, the Red384 has 16, and the HDX has 96 channels. Next, I would like to show you how some of our BLI platforms operate based on different features and throughput capabilities. What is shown here is the interior of the Red96E system. It has an optic box where detectors and the light source is housed, a sample plate where all your experiment samples are housed, and the biosensor tray where the biosensor for a particular experiment is racked. Unlike traditional SPR-based platforms, Octet does not use fluidics to route samples to biosensors. In Octet workflows, the biosensors are mounted to the channel head and moves to the plate, and a dip and read measure collects binding responses. Since liquid does not travel out of the plate at any point, there is no concerns of clogging and enables analysis of samples that are in complex matrices such as lysates, serum, and plasma. And that lets you cut down on your sample preparation times by avoiding steps such as degassing, filtering, centrifugation, etc. Also, Octet platforms are walk-in ready systems where no instrument cleanup or preparation is needed before or after the experiment, help you get more experiment done in a day. The number of channels in the instrument dictates how many biosensor data points can be collected simultaneously. So the Octet Red96E system can collect data from up to 8 biosensors in a single dip and read measure, and complete collecting data from an entire 96-well plate in 12 dips, for example. Next, I would like to move on to the next Octet Red384 that has 16 channels, read head, where you can read up to 16 biosensors, data from 16 biosensors simultaneously. The Red384 system allows you to have 2 sample plates that can either be 96 or 384 well plates. This provides you with the capability to have more samples to be analyzed in a single unattended run, and also less volume when using 384 wells. The Octet Red384 systems are compatible to automation with robotics and liquid handlers. to further extend unattended run times for projects and experiments with larger sample volumes. The last system I want to talk about in my introduction is the Octet HTX, our highest throughput system. The HTX has a user-selectable read head where users can opt for simultaneous analysis of 8 to 16 biosensors that will read at a maximum sensitivity, or opt to use the 32, 48 read heads, or can go to top throughput with the 96-channel read head. At the highest throughput, a single-step immunoassay experiment or a full-plate affinity ranking can be done in minutes. Similar to the Red 384 system, the HTX can also house two 96 or two 384 well plates and perform complete walk-away ELISA assays. Like the Red 384, the HTX is also compatible to integrating with robotics and to extend unattended run times as well. Core to the BLI technology is the biosensor. The Octet biosensor consists of a fiber optic attached to a polypropylene hub with a specific chemistry available at the end of the biosensor tip that will help in ligand immobilization. A typical Octet experiment would involve immobilizing one binding partner to the biosensor before the biosensor reads for binding from the sample plate with test analytes. The biosensor is capable of analyzing binding interactions from a wide range of crude matrices. Some of them are shown here, for example, such as soil, pollen, fuel extracts, or biomatrices such as cell lysates, media, serum, and plasma. Once again, this capability offers users the option to capture ligands from lysate and media, reducing timelines to obtain data. Let's take a look at how the Octet platforms detect binding interactions. The technology is based on biolayer interferometry, where the light is traveling from a light source. It travels through the fiber optic in the biosensor to the highly reflective layer at the end of the biosensor. So when an interaction does happen, what happens is the thickness of the biolayer does increase. So the light wavelength now reflects off of a slightly longer thickness that results in an interference pattern. And then that real-time measure provides you the on-rate kinetics. So right here, the curvature of this nanometer shift response will give you the on rate. And when you dissipate that complex, which we'll call the dissociation rate will provide you the off rate. This is off-the-shelf biosensor chemistries we have to offer that will help immobilize one binding partner to the biosensor. For example, if you want to immobilize antibody-based ligands, one could use FC capture chemistries based on the species, or you could use Pro-A, G, or L-based biosensors. Also note that we have biosensors recommended for kinetic and quantitation applications, and some biosensors are recommended for both. For immobilizing recombinant proteins, one could use our histag capture biosensors, GST, or streptavidin-based chemistries that would immobilize biotin-labeled ligands. We also have biosensors that can immobilize ligands based on traditional amine reactive chemistries or hydrophobic-based adsorption using APS biosensors. We also have to offer several kits that is used for quantitating contaminants such as residual protein A, host cell proteins, or we have a GlyS kit for sialic acid glycan screening. Overall, Octet platforms can offer a range of applications, including kinetic characterization that involves the measurement of binding on rates, off rates that provides affinities. One could also use the Octet for quantitative applications such as immunoassays for biologics quantitation, where users can adopt a direct one-step, a sandwich, or a multi-step assay format to achieve sensitivities from milligrams per mil to picograms per mil sensitivities. And also can be used for screening type assays and as a tool where binding data can be used as part of a hit selection campaign, tests for functional activity of a biomolecule, epitope binning, or a rank ordering experiment. As a result, Octet platforms can be utilized in a range of workflows and programs from target ID, lead ID, and optimization, and all the way to process development manufacturing, supporting these workflows as a kinetic characterization assay, a diversity assessment such as an epitope binning experiment, or as a quantitative or potency assay. all of the platforms and software can fully support processes in GMP-based environments as well. With that, I would like to hand over to Dr. Wei-Hong Chen to talk about his work on SARS-CoV-2 vaccine development. Dr. Wei-Hong?
Hi, good morning, everyone. Yeah, I'm Wei-Hong Chen from Walter Reed Army Institute Research, the Emerging Infectious Disease branch. So yeah, today I'm going to share our effort to develop a vaccine to the current SARS-CoV-2 pandemic. The topic is the rapid characterization and the selection of a SARS-CoV-2 vaccine candidate by biolayer interferometry BLI technology. So in our structural biology lab, and we are capable to utilize tools we are good at, such as the crystallography, protein immunogen design, antibodies engineer, and the different assays to understand the threat that we are facing in the environment. But today, that is the SARS-CoV-2. In addition, we learn the lessons from our immune system, such as the immune response and the other our physical reaction. The goal is trying to find a cure or vaccine to eliminate this threat from the human. So here's the overview for today. I will briefly start with what is the coronavirus and its background, and then introduce my research that includes the projects I used the Octet, and then the summary, and then my acknowledgement. So for the current COVID-19 disease, as you may already know, comes with these symptoms from the head to toe, and sometimes it could get worse with the complications. Here's the slide shows the burden of COVID-19 to date. Almost 17 million people are infected, 1 million people is hospitalized, and 661,000 were dead because of this COVID-19. Although the mortality rate of the COVID-19 is not very high compared to other viruses, but considering so much population are suffering from the disease in a very short time, it is still a very serious number. And not only individuals are affected, the economy and society is under the burden of this pandemic as well. We have direct impact of the lockdown and also society impact as well. So the coronavirus, of course, are responsible for this pandemic, but not all the coronaviruses are this lethal. Coronaviruses are a group of RNA viruses that cause diseases in mammals and birds. The bat could be the natural reservoir for the coronavirus, and the virus could be transmitted to other animals such as the civet or camels, and then eventually to the human. As we know for now, 7 strains of the coronavirus can infect human. In humans, this virus causes the respiratory tract infection that can range from mild to lethal. 4 human coronavirus circulated in the human population and produced the general mild common cold symptom in adults and the children worldwide. However, 3 human coronavirus causes the outbreak and produce potentially severe symptoms. These are the 2003 SARS, and the 2012 MERS, and the current COVID-19. Today, I'm going to focus on the SARS-CoV-2, of course, that is for today's talk. And on the left is the cartoon picture illustrate the SARS-CoV-2 virion. The major protein on the surface is the spike protein or spike glycoprotein that is responsible for the interaction with the host cell. The atomic structure of the spike glycoprotein is solved and that could be found in protein database. It is a trimer with the shape like a mushroom. It is big, more than 400 kilodalton in this trimer configuration, and it is why it is visible as a crown under the microscope. On the right is the top view of the this trimer spike protein. The spike protein plays an important role in the infection process to the host cell. The spike protein interacts with the human S2 receptor with its receptor-binding protein or receptor-binding domain. During the process of a configuration of the trimer spike change, the receptor-binding domain or RBD splits up while interacting with the human receptor. This is one of the critical steps allowing the virus to infect and entering the host cell. And it is also reported if we could target the RBD of the spike, it is possible to limit the infection process. In this slide is the flowchart for our vaccine development. In our lab, we are focusing on the protein-based vaccine, and we use the structural information to design our protein construct, and the recombinant protein is expressed in mammalian cell line and purified to homogeneous. These vaccine prototypes are categorized by several methods in order to downscreen to enter animal studies. The animal studies result is also detailed categorized and evaluated for further preclinical and clinical research. So I'm going to share some of our research efforts towards SARS-CoV-2, starting from the structural information to antibody binding to the antigen, and then development of the vaccine in rare and EIDB department. So our EIDB, the emerging infectious disease branch, has been working on the coronavirus for a while. We have the first MERS human vaccine trial in 2018, and then the phase 2 of this MERS human vaccine study is planned in 2020 and 2021. So on the January 11th, the sequence of SARS-CoV-2 genome was released, and we began to work on the vaccine design. On the January 23rd, we ordered the antibodies from the BLI resource, and right after receiving the antibody, we determined the CR3022 and two antibodies, CR3022 and the 24CD binding affinity on the January 30. So when we compare the DNA sequence among the coronavirus, SARS-CoV and the SARS-CoV-2 share almost 80% of the genome is similarity. The sequence differences is shown here on the RBD structure in red. We realize the differences is mostly located on the loops of the RBD, and actually the SARS-CoV-2 spike protein sequence are even more similar to some spike protein than the SARS coronavirus. So we quickly identify a few monoclonal antibodies that bind to the SARS, can also bind to the SARS-CoV-2 receptor binding domain. One is the human antibody, CR3022, and another is from the mouse, 24CD. Not surprisingly, these antibodies also bind to the bad coronavirus, shown here on the right down corner. The binding affinity of these two antibodies, CR3022 and 24CD, is pretty strong. The KD, the dissociation constants of these two monoclonal antibodies are measured by the octet, and they are around 10 nanomolar range. Now we know these two proteins are binding to the SARS-CoV-2 RBD, and we perform the competition assay to identify if they bind to the similar epitopes. In this assay, we use the His1K probe to pick up the RBD protein, which has the His tag on it, and sequentially dip into monoclonal antibodies. So I'm showing here the competition data among 3 antibodies in this slide. It turns out these antibodies, the 3 antibodies, CR3022 and the 24CD, and also the SB1, are competing each other. And the CR3022 Fab complex with the RBD protein, the crystal structure of it was solved, and the CR302 epitope is highlighted here in the purple. The epitope area are highly conserved between SARS-CoV-1 and SARS-CoV-2 on RBD, this protein. This may explain these 2 SARS antibody has the cross-reactivity to SARS-CoV-2 Spike glycoprotein. Although we know the CR3022 epitope is different from the human S2 receptor-binding interface based on their SARS model, I still want to assay the competition between antibodies and the human S2 receptor in order to see if any conformational change after the antibody binding, antibody bound on RBD would interfere in the binding of the human S2 receptor. So the similar strategy is used here for this assay, but the results show there's no competition or inhibition for among these antibodies and the human S2 receptor. In the past few months, our lab is working very hard to produce protein-based vaccine. We use the structural information to design our protein construct. Within 3 months, we have designed, expressed, and purified more than 200 constructs. All of these vaccine prototypes are characterized by several methods in order to downscreen to enter animal study. Today, I'm going to show a few of our vaccine constructs that I characterized with ArcTech, and some other data to support the vaccine meet our expectation. So in this slide, I'm showing our protein vaccine design. On the left is the model for our vaccine. We name it SPFN, Spike protein ferritin nanoparticle. In the center of the ferritin, here, in the center is the ferritin protein particle, and the spike protein are as a trimer engineered on the ferritin particle surface. There are 8 trimer proteins on the surface of ferritin. In the middle shows the size exclusion chromatography purification. The major peak we are expected as a nanoparticle. On the right panel of this slide, the negative stand EM is shown here with the 2D class average. It indicates the spike on the ferritin particle and the dimension of the particle is close to a calculated model. Since we have purified a protein, we want to and we want to understand if it is properly folded. So the ArcTech is used to test the binding between antibodies and our vaccine candidates. Over the time, we also isolating a set of antibody that it binds to SARS-CoV-2 Spike protein, so we can have more information from this set of antibody. Here we loaded antibody to AHC probe and then dip into our vaccine candidate, shown on the right. In this slide, I'm only showing the SPFN1 binding curves of the different antibodies. Using the ArcTech, it is efficient to characterize our vaccine candidates, and because it is label-free and also it runs pretty quick. And we can have real-time measurements of the binding, and we can recover our samples for other experiments, such as we can take the samples direct from the well and put it onto the electron microscope grid, and then to see under the microscope. So it is not only we can check the quality of the protein, but it can also measure the quantity. Shown here with the two different antibodies with the different concentrations. So based on the curve we get from the octet, we can make a standard curve, and then we can know how much protein we purified in the final. So after characterizing hundreds of our vaccines, we chose 28 prototypes into a mouse animal study. We collected the mouse sera over the experiment with the different time points. 3 sera samples were collected in 2 weeks after immunization with our vaccine prototypes, and characterized this sera with using our recombinant proteins. Specifically, we are looking into the reactivity to the RBD binding. Since the advantage of the octet, that is, can assay the mixture sample, we always assay the sample on the day we collect or process the sera sample. We tested 100 of mice sera, but here I'm only showing 3 groups of our vaccine prototypes. So here's the summary of this mouse study with the 2 different strains. We used the BOB-C mice and also the C57 BL6 mice. As you can see here, in most of the mice, the RBD-specific antibodies are elicited after the second immunization, and the response is increasing after the third immunization. It gives us a good confidence that our vaccine meet our expectation to induce the immune system to produce the antibody that we want in animal study. So I would like to come back a little bit to talk more about spike and the human receptor recognition. It has been reported that spike protein and the human receptor recognition is the first step in viral entry to the host cell. The receptor binding domain is responsible for direct binding to the human S2 receptor and trigger the following reactions downstream. Based on this, if an antibody targets the interface of the RBD and the S2 receptor, it has a great potential for neutralize or to limit the SARS-CoV-2 infection. So, do we have the antibody targeting in the interface from the sera we collect in our mouse study? In order to answer this question, we developed an human S2 inhibition assay. We have a modified strategy as competition assay, but assays with the serial dilutions and calculate the human S2 binding signal. Shown here are the results. As you can see, with the different dilutions, the inhibition of the S2 binding is decreased. It indicates the sera do have the antibodies that targeting the receptor binding domain and the S2 receptor interface. So here's the quick summary. We identified the monoclonal antibody with the cross-activity toward the SARS-CoV-2 and the SARS spike protein using the octet and the antibodies obtained from the BLI resource. And it is very efficient to characterize more than 200 prototypes of the vaccine candidates within 3 months. And we also evaluate this vaccine, the sera from the animal study and against the receptor binding domain. we also use this S2 inhibition assay to understand the antibodies targeting on the receptor binding domain area. So here I'm going. So here I'm thankful for our lab everybody was so hard to finish to go to this stage, and also the rare other labs, they give us a lot of support to work together to study this COVID-19 pandemic. And the collaborators from NIH, NCI, and Fabric, they screen a lot of EM data for us, and AstraZeneca providing several antibodies that targeting on the SARS-CoV-2 Spike protein, and also USU Uniform Service University also cooperating with us. Yeah, thank you very much.
Thank you, Dr. Wei-Hong, for the really nice presentation. So we have a few questions that came through while you were talking. So let's go over a few. So question number one is, do you need to include a trimerization domain example in your construct to form ferritin nanoparticles, or other stabilizing mutations are necessary?
The mutation is necessary on the spike protein to stabilize the trimer and also the ferritin nanoparticle. And that is why we have so many different constructs. Yeah, we look into the structural detail to figure out, and also to figure out the stability and making the mutations to prove our design is work. So yeah, it is definitely need to mutate the protein to stabilize this nanoparticle vaccine.
Okay, is that so? The picking of the right candidates, was that somehow related to binding signal or binding affinities? Is that another way to look at recognition?
Yes. So basically, we have several things to check the good nanoparticle or bad nanoparticle. First is the expression level. If the expression level is not good, yeah, we don't think it is a good candidate for the vaccine. The second one is how good this nanoparticle looks like under the microscope. We do want to see good nanoparticle. I'm showing here on the slide. We have spikes on the core of the ferritin. We have several constructs that have loose spike protein, and we don't think that is very good for the further down. And then the third one is the octet. So antibodies bind to this spike protein. Even it is a well-deformed trimer, but if the antibody doesn't bind to it, we don't consider it is a good candidate for our vaccine.
All right. The next question is, I saw you use the His1K for analysis. How is the sensor? Do you regenerate the biosensor?
We don't regenerate the biosensor. Yeah, we just use it once and just get rid of it.
Okay. Which expression system do you use to produce the nanoparticle?
So we use the mammalian cell line, SP293 cell line to produce this protein.
Next question, where you got the His-tagged receptor binding domain from? Did you express it or got it commercially? I guess that's the question.
Yes, we express in our lab as well.
Okay. Next question, how many disulfide bonds are in the SARS-CoV-2 protein, majorly in the receptor binding domain?
How many? I don't count them up. I don't count as yeah. I need to go back to check it. Yeah.
Sure. We can get back to the answer with that. The next question, when you said you quantitated, use the octet as a quantitative method, is that similar to an MSD or any traditional quantitation experiment?
I think the idea is similar, because the different concentration gives a different intensity of the signal. So as long as your signal is reliable and can produce the standard curve, it can be applied to this method.
Okay. Next question. Slide 20 had a very short baseline after loading the nanoparticle. Any specific reason for that?
Slide 20 have a very sharp baseline for short baseline.
Yeah.
No, there is no specific reason. I just want to go into a baseline, kind of wash away the unbounded antibody, and then go to the antigen.
Okay. Next question. Have you challenged any mouse or any other animal subject with COVID to see if the vaccine candidates provide protection?
We are planning to do it. There is ongoing.
Okay. So those seem to be the most questions that we've gotten. So any other questions from the audience? We have a few minutes, or if you think of any questions to either myself or Dr. Wei Hong, you have my email address and my contact information in the slide that is visible right now. Please don't hesitate to ask. We will get back to you. Oh, we're getting a couple more questions. Can I directly immobilize the S-trimer protein on the chip and check its binding with the antibody?
Yes and no. Yeah, so if you want to immobilize the protein, you need to have the tag on it. Or you need to use the chemical immobilization kit. I think the Forte Bio have the kit and do the chemical immobilization, and then you are ready to go into the antibody.
All right. If there's no more questions, I would really like to thank Dr. Chen for his beautiful talk and his efforts in the coronavirus to support and help us find a cure to this pandemic that has paralyzed all of us in known and unknown ways. So, and we would like to thank and wish them good luck in their research. And we expect to see a cure to the vaccine soon, even from the efforts from this lab and his colleagues. Thank you very much for the presentation, Dr. Chen, and everyone who attended this presentation. Thank you very much.
Yeah, thank you very much.